My name is Alexia Ciarfella, and I'm a food science and nutrition double major, and my project is translating research on the luminescence of grass probes into educational tools for photophysics. My project takes research that has already been done in our lab on the luminescence of food molecules, particularly artificial and natural dyes, and it translates it into science experiments for kids. In this experiment, the kids will make about 4 different types of candy that have different water contents. So they'll range from soft to chewy to a hard candy like a Jolly Rancher. And each of these different candies has a different molecular matrix. And in quenching, when you change the matrix, you change the intensity of the fluorescence. So as the matrix hardens, or if you go from a Starburst type candy to a Jolly Rancher type candy, the food will fluoresce more. So this is because when an electron gets excited, it can progress down to its less excited state by either non-radiative or radiative decay. Non-radiative decay happens when it moves around and emits energy through that way. Or radiative decay is when it can't move around and has to emit a photon to get rid of its energy. So in a tougher matrix, like a Jolly Rancher matrix, it emits a photon because it doesn't have a lot of space to move around. But in a Starburst type candy, there's more space to move around because there's more water activity in its hydration shell, and it doesn't emit as much light. Another experiment I'm working on is called Rainbow Ghosts, and it uses a diffraction grating, which separates light out by wavelength, and you tape it over the lens of a camera phone and shine an LED light on a food source, and on the screen of the camera phone comes up these awesome rainbows that show what wavelengths of light the food emits. This experiment helps teach kids about the lab machine called a luminescence spectrophotometer, which we use almost every day, and it might seem big and scary to them, but seeing it on a cell phone screen makes it a lot more accessible to them. My research is a little non-traditional because it's more of applied and educational science rather than exploring new concepts. What has surprised me the most about research is that I've had to interact with people more than I thought I would have to. For instance, a few weeks ago I went to the National Institute of Food Technologists conference in Chicago, and my PI told me that I needed to talk to different color companies in order to get some samples. So I had to network with different color houses and contact them via email in order to get samples for our lab. I got involved through the Rusty Summer Science program because one of my friends from the SEBS Honors program suggested that I apply for it, and once I looked into it, I knew it was the perfect program for me. I work under Dr. Ludischer, and I work closely with Dr. Maria Corradini. Working with them has been great because they're some of the smartest people I've ever met. For instance, if I come to them with a problem about my research, they can give me about 10 solutions. My research is important because it inspires a whole new generation of scientists. It also introduces them to concepts that they'll use later in their career, particularly spectroscopy, which is used in all sorts of areas of research from genetics to microbiology to food chemistry, like my lab.